In question 68, we're told that a student carried out an experiment to investigate the effect of enzyme concentration on the rate of an enzyme control reaction. We're given this diagram of what the student did. He used a starch agar plate and 5 wells, shown here in the key. These black circles represent a well in the starch agar containing different concentrations of amylase solution. The starch agar plate was then incubated overnight, and then the plate was flooded with iodine, and what was found was that there was an area around each of the wells which was partially digested and therefore did not stain with iodine, and we're told the diameters of those regions. So we have 27 millimeters for the 1% amylase and 9 millimeters for the 0.001% amylase. So you can see there's a general trend of increasing as concentration increases. But beyond that, we're asked which of these three factors could have affected the result. So concentration is there, but before that, let's talk about pH, since it's number 1. Could pH have affected these results? Well, let's think about it. Amylase is an enzyme which digests starch. How do enzymes work in relation to pH? Well, there's normally an optimum pH at which enzyme activity is at its greatest. Beyond this point, any increases or decreases in the pH will affect the hydrogen bonding between the polypeptide chains in the tertiary structure of the protein, the enzyme being the protein, and therefore it would affect their capacity to form bonds with the substrate, and therefore enzyme activity would reduce. So pH affects enzyme activity, therefore pH of starch agar could have affected the results. Let's move on to concentration. As we can see from this graph, enzyme concentration normally does affect activity up to a point of saturation, beyond which there is a maximal number of enzyme substrate bonds, and any increases in enzyme activity any increases in concentration of enzyme will not have an effect, since the concentration of substrate is the limiting variable. Now, if we have a look here, there is a continuous trend of increase which corresponds to the increasing arc of the graph. So we can say, yes, concentration of amylase does affect enzyme activity. So both of these are right so far. We can mark that on. And now let's move on to temperature. Again, temperature, of course, does affect enzyme activity in that there is an optimum temperature above which there is the danger of denaturing as the bonds between the polypeptide chains in the tertiary structure become weakened and break, therefore changing the shape of the active site of the enzyme, affecting its capacity to bond with substrate. But at lower temperatures, there is a lower kinetic energy and therefore less total enzyme activity. So yes, temperature could have also affected the result. So since all three could have affected the result, we know our answer to be H.